Good morning, students. This is class 9, subject science, chapter 5, the fundamental unit of life, lecture number 6. Now, in the previous lecture, I discussed various cell organelles like mitochondria, plastids, and vacuoles. Now, this is the last topic of this chapter, that is plant cell and animal cell. Now, what is a plant cell and how the diagram is there? You can see from the diagram that plant cell, it is rectangular in shape. The plant cell is rectangular in shape. There is a cell wall that is present outside the cell. There is a cell wall which is present outside the cell, and this cell wall, it is made up of cellulose. The cell wall, it is made up of cellulose, and this cell wall, it is present only in plant cell. It is absent in animal cell. I discussed this point while I was discussing about cell wall. So the cell wall, it is present in plant cell and it is absent in animal cell. Now after cell wall, there is a covering cell membrane. What is the other name of cell membrane? Plasma membrane. So this cell membrane, it is present both in plant cell as well as animal cell. So first is cell wall, then second is cell membrane. Now you can see in the center, there is a white colored part. This white color part is actually vacuole. What is this? This is vacuole, and this is very large and it is centrally located in case of plant cell. But in case of animal cell, the vacuole, it is located at the periphery of the cell. It is located at the periphery of the cell and it is small in size as compared to this plant cell vacuole. Okay. Now after vacuole, you can see there is a blue shaded part that indicates nucleus. The nucleus is present in plant cell, and inside the nucleus there is a darkly stained structure that is called as nucleolus. Now this nucleus, it is surrounded by endoplasmic reticulum. You can see the yellow part from here. It is endoplasmic reticulum. Rough endoplasmic reticulum and smooth endoplasmic reticulum. Now rough endoplasmic reticulum, there are some black structures present which are called as ribosomes. So that is why the surface is rough. But on smooth endoplasmic reticulum, there is no ribosome present. So that's why that is called as smooth endoplasmic reticulum. Now are you clear about the endoplasmic reticulum? Okay, now coming to next cell organelle, Golgi body. You can see this orange shaded part where the vesicles are stacked together in layers. These are called as Golgi bodies. Now beside Golgi bodies, there are chloroplast present, a type of plastid, and there is also amyloplast present. This amyloplast, it is also a type of plastid that I have not discussed with you, but it's your wish to write amyloplast or not, it hardly matters. So it's better if you write chloroplast because it is there in your syllabus. So chloroplast is present, that is a type of plastid. Now beside chloroplast, you can see there is mitochondria. In mitochondria, there are two membranes, inner membrane and outer membrane. So this is about plant cell. Now come to next, animal cell. In animal cell, there is no cell wall present. There is no cell wall present. But in case of plant cell, it was present. Now cell membrane. You can see in the animal cell, there is a cell membrane present. What is the other name of cell membrane? Plasma membrane. So the cell membrane or the plasma membrane is present in animal cell. Now one more thing. The plant cell, it was rectangular in shape, whereas this animal cell, it is oval in shape. What is the shape of animal cell? It is oval in shape. Now cell wall is absent and cell wall is present. Now what about the vacuole? In case of plant, it was large and centrally located. But in case of animal, it is small and it is present at the periphery of the cell. That is green part. That is vacuole. That is shown in animal cell. Now you can see there is a nucleus, blue shaded part. That is nucleus. Inside the nucleus there is nucleolus, as like there was in plant cell. Right? Okay. Now beside nucleus, you can see endoplasmic reticulum, rough endoplasmic reticulum and smooth endoplasmic reticulum. In rough endoplasmic reticulum, the surface is rough, and in case of smooth endoplasmic reticulum, the surface is smooth. In rough, ribosomes are present. In smooth, no ribosomes are present. Now another organelle you can see mitochondria, that is also present in animal cell, and lysosomes. These lysosomes, suicide bags, they are also present in animal cell. Golgi apparatus, Golgi vesicles, they are also present in animal cell as like it was present in plant cell. So this was about animal cell. So now what is the difference between plant cell and animal cell? It is larger in size. Animal cell is smaller in size. Plant cell, it is rectangular in shape. Animal cell is oval in shape. In plant cell, cell wall is present outside the cell membrane. In animal cell, cell wall is absent. In plant cell, a single large centrally located vacuole is found, and in case of animal cell, vacuoles if present they are smaller, many in number, and they are scattered in the cytoplasm. In case of plant cell, nucleus is found at the periphery. In case of animal cell, nucleus is found at the center. In case of plant cell, plastids are found, and animals plastids are absent. This is a very important point. Write down. Plastids are present in plant cell and they are absent in animal cell. Exceptions are there, protozoans and euglena. Now in plant cell, the stored food is in the form of starch, and in case of animal cell, the stored food is in the form of glycogen. So now you can differentiate between plant cell and animal cell. So this ends for today's lecture. Now I would be starting the summary of the whole chapter. Now if any student has any doubt in any of the topic, you can see the summary. So what is cell? Smallest unit that carries out life's activities, that is cell. Now group of cells, they work together, they form tissue. Then group of tissue work together to form organ. Organ, group of organ work together to form organ system. And that organ system finally forms an organism. So this is how the organization of life is there. Now this organism, it can be made of one cell or many organ systems. Now cell theory. This is a very important theory. It was given by Sheldon and Schwann in 1838 and it was further extended by Virchow in 1855. Now the cell theory states that number 1, all the plants and animals are composed of cells. Number 2, cell is the basic unit of life. Number 3, all the cells arise from pre-existing cell. This is a very important statement. All cells arise from pre-existing cell. Now what does this statement means? It means that every organism starts life as a single cell. So this was the cell theory. Now types of the organisms on the basis of number of cells. There are 3 types of organisms: non-cellular organisms, unicellular organisms, multicellular organisms. Now first is non-cellular organisms. Non means not even a single. Cellular means cell. So those organisms which do not contain any cell in their body organization, those organisms are called as non-cellular organisms. Now what is the example? Virus. Second is unicellular organisms. Uni means single, cellular means cell. So the organisms which contain only single cell and all the functions they are performed by that single cell, and as a result its lifespan is short. Examples are amoeba, paramecium and chlamydomonas. Now multicellular organisms. Multi means many, large number. Cellular means cell. So those organisms which contain many cells, which contain large number of cells, these are called as multicellular organisms. Now many cells are present means different cells perform different functions and as a result its lifespan is long. Now what are the examples of multicellular organisms? Human, trees, animals, etc. So this was the classification of the organisms on the basis of number of cells. Now moving to next topic, types of solutions on the basis of concentration. So there are three types of solutions. Number one, hypertonic. Number two, isotonic. Number three, hypotonic solution. So let's discuss one by one each. Hypertonic. Hyper means more. Isotonic. Iso means same. Hypotonic. Hypo means less. So first is hypertonic. It is that solution in which more concentration of water is present inside the cell. I am repeating again. Hyper means more. So more concentration of water is present inside the cell. Just consider inside the cell only. Otherwise you will get confused. And consider only solvent. Two things. Solvent and inside the cell. So hyper means more amount of water present inside the cell. Now more amount is present inside the cell and outside the cell it is less. Now we know that water moves from high concentration to low concentration. So water will move from inside to outside. So the cell shrinks. Isotonic means same concentration of water inside the cell as well as outside the cell. So there is no net movement of water across the cell membrane. So the size of the cell is normal. Now hypotonic, hypo means less. So less amount of water, and where? Inside the cell. Now less amount of water is present inside the cell and outside the cell it is more. So water moves from outside to inside. So the cell swells. Example, raisins. Raisins means kishmish. When raisins are kept in water, they swell up. This is due to hypotonic solution. A very good example of this. Now next topic is difference between cell wall and plasma membrane. Cell wall occurs in plant cells. Plasma membrane or the cell membrane, it occurs in both plant cells as well as animal cells. Cell wall, it lies on the outside of the cell, whereas plasma membrane, it lies on the outside of animal cell and inside in plants. Cell wall is rigid and thick. Plasma membrane is flexible and thin. Cell wall is permeable. Plasma membrane is selectively permeable. Note down these two terms. I discussed in the first lecture also. Selectively permeable and permeable. Cell wall is formed of cellulose. Plasma membrane is formed of lipids and proteins. Cell wall provides protection and strength to the cell, and plasma membrane, it regulates movement of molecules inside and outside the cell. Now you can see the structure of nucleus. There is nuclear membrane, that is a membrane that surrounds the nucleus. Now that nuclear membrane contains pores. What are those pores called? Nuclear pores. And what is the function? What is the function? To allow the movement of the materials from the nucleus to the cytoplasm of the cell. Now inside the nucleus there is a fluid called as nucleoplasm, and there is a dense spherical structure that is called as nucleolus. And there is also one more thing, chromatin, thread-like structures which are present inside the nucleus. So this was the structure of a nucleus. Now what are prokaryotes and eukaryotes? Prokaryotes are the organisms which lack nuclear membrane. They do not have membrane-bound cell organelles. Example, bacteria, cyanobacteria. And what are eukaryotes? They have nuclear membrane. Also, they have membrane-bound cell organelles. Example, plants, animals, and fungi. This is a very important point. Now, I will be discussing various cell organelles in brief again. What are those various cell organelles? Endoplasmic reticulum, Golgi apparatus, lysosomes, mitochondria, plastids, and vacuoles. Now first is endoplasmic reticulum. What are the types? RER and SER. RER means rough endoplasmic reticulum. SER means smooth endoplasmic reticulum. Now RER, rough means surface is rough. Now why rough? Due to ribosomes. Now what is the function of ribosome? Synthesis of proteins. SER, smooth, surface is smooth. Why smooth? No ribosomes present. And function, synthesis of lipids and fats. Yes, students, note down one more thing. Proteins or ribosomes. There is a connection here. Ribosomes are called as protein factories of the cell because ribosomes are related to the synthesis of proteins. This may come in 1 mark. Now next is Golgi apparatus. It was described by scientist Camillo Golgi. What is Golgi apparatus? It consists of a system of membrane-bound vesicles. Vesicle means sac-like structures. They are arranged parallel to each other in stacks which are called as cisterns. So this is a diagram of Golgi apparatus. You can see from the diagram that from Golgi apparatus, primary lysosome is formed. So you can write down the function of Golgi apparatus: synthesis or production or formation of lysosome. One of the function of Golgi apparatus. Now next is lysosome. What are lysosomes? These are a kind of waste disposal system of the cell. How? They help to keep the cell clean by digesting any foreign material as well as worn out cell organelles. Okay, they help to keep the cell clean. Now there is a question, why lysosomes are known as suicide bags? The lysosomes are known as suicide bags because the lysosomes, they contain digestive enzymes inside them. They contain digestive enzymes. Now, whenever there is any disturbance in the cellular metabolism, what happens? Inside the cell lysosome, these lysosomes, they burst. When the lysosome bursts, the digestive enzymes which are present inside the lysosome, they will come out and they will digest the cell. So in this way, it helps to keep the cell clean. So this is the reason lysosomes are known as suicide bags. Now you can see mitochondria, inner membrane, outer membrane. The inner membrane is folded into finger-like projection. These are called as cristae. And what is matrix? Matrix is the fluid which is filled inside the inner membrane of mitochondria. Now mitochondria, they are known as powerhouse of the cell. Why? Because mitochondria, these are the site of cellular respiration. Means where cellular respiration occurs. Now the food is oxidized, and when the food is oxidized, energy is released in the form of ATP. Full form of ATP? Adenosine triphosphate. Now energy is released in the form of ATP when the food is oxidized. Now this energy, it is used for making new chemical compounds. So that is the reason mitochondria are known as powerhouse of the cells. Now the next is plastids. What are the types of plastids? 3 types: chloroplast, chromoplast and leucoplast. Chloro means green. They are green colored plastid. Now why they are green? Because they contain a pigment called as chlorophyll. And what is the function now? Photosynthesis. Very clear. Chromoplast. They are colorful. They are colorful plastids except green. And they are present in flowers and fruits. You might have seen the flowers and fruits, they are colored. Why? Because chromoplasts are present inside them. Now what is the function of chromoplasts? They help in pollination. And the last is leucoplast. Leucoplast, they are the colorless plastids and they help in the storage of starch, oil, lipids. The last topic is vacuoles. What are vacuoles? These are the storage sacs for the liquid or the solid contents. They are bounded by a membrane. That membrane is called as tonoplast. They have a fluid filled inside That fluid is called as cell sap. It contains sugar, amino acids, proteins and minerals. In animal cells, vacuoles are small size, and in case of plant cells, vacuoles are large in size. This is a very important point. Note down. So what is your homework for today? You will do question number 1, which is given on page number 66 of chapter 5 NCERT. And the students who are not having the books right now, I am giving the question. You can write down in your notebook. Make a comparison and write down ways in which plant cells are different from animal cells. Now, I would be discussing the homework solutions of the lecture 5, means the answers of the questions which were given in the previous lecture. Can you name the 2 organelles which we have studied that contain their own genetic material? Answer: mitochondria and plastids. Which organelle is known as powerhouse of the cell and why? Mitochondria. Why? Because these are the sites of cellular respiration. They release energy in the form of ATP, which is used by the organelles to carry out the basic functions.